unit. It's really to focus on all of the rest of the application. And I can't emphasize enough that sometimes the rest of the application, it's sometimes even longer than the main grant application. And there's a really strong tendency for people to be so wrapped up into the science that they forget the rest. And sometimes, even when I've reviewed grants, it's very clear that all of the remaining parts of the application were done at the last minute, very, very slipshod. And they're actually very, very important.
You don't know if that's okay for you. I like it.
So they're actually very, very important, and that you can't leave them to the last minute. I usually actually advise that even when you start to think about putting together an application, that's when you really need to figure out what's needed for the rest of the application outside the application. And it's also work on it as you go along, because from human nature, you're going to be wanting to work on that research plan and polishing it perfectly up to the last minute. The last thing you need is a research administrator saying, you need to write these 5 pieces or I can't submit your application for you. So, and with that, the very first thing, and you can, hopefully people can see the PDF that's up, is the first most important thing is work with your research administrators. And that's giving them really far in advance notice that you're submitting. And as soon as possible, the second you think you're going to write a grant, is to get with the proposal. Now, many people who are on this call are truly new investigators. There are lots of things that have to be done even before you're eligible to submit, such as put together your NIH Commons ID, and so forth and so on. And if you've never submitted or really interacted with for instance, the National Institutes of Health, you need to put that together. Similarly, things for the NSF you have to put together, as well as this is true for pretty much any other agency. And also that even as you're sketching out your specific aims, where you're really trying to figure out what you're going to propose research-wise, you really need to figure out what the non-research plan information you need to submit. And you need to familiarize yourself with those directions, and you need to work on them early. That depending on the type of grant, there can be things that need to be approved many, many months out. Now, NIH has a policy of what's called just in time. There are certain things that you don't have to get approved way far in advance, that you have to get approved though before they give you the money, such as human subjects and animal subjects approvals. However, you still almost need to have those written because you have to submit the draft documents into the application. So you, again, can't leave that stuff for the last minute. So most of what I'm going to be focusing on here is related to the NIH. That's what I have the most personal experience with. However, every grant application has these components. They're just done differently. I'm in the middle of submitting a grant with the Indian government, with an investigator in India right now. And all of those same pieces are in that grant to the Indian government. It's just what they call them are different. So these are things that you really need to take into account. Okay, so the first part that I really want to talk about is the environment page. And the thing about the environment pages is that they are probably the piece that is the most commonly short shrifted in applications. And people will put in things that are clearly institutional boilerplate, or they'll just write a very minimal description. Now, to be perfectly frank, this is a human nature sort of thing. If we were all investigators at the Harvard Medical System, or at Johns Hopkins, or at UCLA, yes, you can get away with writing very minimalistic canned boilerplate environment pages. When somebody sees where you work, they're like, yeah, sure, their environment's great. Not going to worry about it. The problem is that, we are in an idea state. All of you who are probably listening to this are either related to the Center for Translational Research or other things institutions throughout Delaware, that, to be perfectly frank, we are not known entities in many, many study sections. I know that in my personal case, that up until about 3 years ago, I was the only person funded in the entire state of Delaware from the National Eye Institute. I think you could probably guess that the grant reviewers have zero idea what the institutional resources are at the University of Delaware. And this is going to be even more so the case at some of the other smaller research institutions that are participating in this. Thus, you really do need to very explicitly, and go ahead and have this be 2-3 pages long, that you need to really think about from the standpoint of your application, what space and equipment you need, what other ancillary things do you need. If you're working on clinical things, you really need to specify the clinical resources. While I may know that Christiana Care is like 4th in the country in number of babies delivered, and I may know that Nemours is a really serious high throughput children's hospital, you can't count on your grant reviewers knowing that, because that it's not like there's this name recognition, your grandmother's heard of it, national recognition of some of these places. So it's really, really important that you do have to spell out what's going on. Also, many of the people who are on this call are new investigators. Study section members are very, very sensitive to people making the transition to independence. And that if you are in a position that could in any way, shape, or form, there could be a question about your independence or your ability to have access to the resources at the Institute, you need to really spell that out as well. So as I say here, it says make both the extent of your dedicated research space. So what space will you have to do this work, as well as the overall environment. And that it's really, you can start with somebody's canned boilerplate. But to be honest, for any of the institutions that people are calling in here, if that boilerplate doesn't start at a couple pages, it is not well developed enough. And then you have to look at that boilerplate and really sell that you have everything you need, whether that is really high-end equipment. Like if you're proposing a high-end confocal imaging aim, you better specify in those environmental pages exactly what instruments you have, what the technical support available is, and the level of access you can have to that instrumentation. If you're working on clinical research things, specify, whether there's research coordinators, specify what the patient population is. I've definitely seen a lot of grants that have a more clinical bent who get basically trashed because the reviewers aren't confident that there is adequate patients or adequate clinical access to be able to collect statistically significant results. So it's really, really important. That I will tell you that my environment pages when I submit to the NIH run about 3 pages. And this talks about all of the major core facilities that I use. I even talk about some of the facilities at Nemours and Christiana Care, because we do have access to things through the entire Excel program and all the core facilities. If you need statistical support, definitely outline all of the statistical support things that are available to you through Excel or CTR or your own home institutions. And talk about, maybe this person has this technical expertise. So it's really critical. And there is actually a section under environment for clinical and for animal. And sometimes I'll just see, oh, there's an animal facility. No, if you work at one of our institutions, don't let that be adequate. It could be you'll get away with it, but what you don't want is for somebody to be scoring the environment at a 5 or a 6, when we all know that we have adequate for most of what people want to do here. We do have very, very strong facilities, but nobody's going to assume that for us. And I think I really can't emphasize that more. Okay. So any questions about environment for right now? Okay. All right, so Biosketch. Now, Biosketch has been a moving target in the past several years. Some of this has been because NIH has now changed the Biosketch format 3 times in the past 6 years. Personally, I'm not real fond of the way they have moved the Biosketch. I really hate it as an NIH grant reviewer, and I don't read most of it. I go directly to the link on my bibliography, but you have to do it. You can't short shrift it. And I think largely that the Biosketches in the new format aren't being used by your grant reviewers. What they're being used by is the program officers and people who are upper in the echelons of the NIH. Like anything here, you're going to be submitting to a variety of grant mechanisms. You have to follow the directions for your grant mechanism. Make sure you do the Biosketch like they say. Yes, it's a pain, but you have to do it. Now, what's really important about the new Biosketch is that it's become a marketing document. In the old days, it was just, this is what my education was, this is what my service is, this is all my pubs in the past 3 to 5 years, this is what my funding is. Now there are these paragraphs where you really have to sell your accomplishments. And it's a happy medium that you do need to really highlight the importance of your scientific accomplishments. However, you also need to be very, very careful not to annoy the reviewer when you're a young investigator that basically says you have cured cancer or something, when the rest of your accomplishments don't support that. So it's really sort of important to write this as a marketing document that really is, again, customized for the application. Now, for those of you who are applying as a PI here, this is your opportunity to say why you have the expertise to actually do the project. And this is maybe one advantage of this format is that you might have published on a technique in 5 publications, but if the reviewer doesn't know that and you propose that technique, they might think that, oh, you're a young investigator, you don't have expertise in that area. In this part of the Biosketch is where you can say, I have published 4 papers using this particular technique, and thus I am well qualified to apply that technique to this new project. One thing to be very, very careful about Biosketches is that for many of you who are trainees, you're going to have to include your mentor's Biosketches. And it will be either your primary mentor's Biosketch or your mentoring team's Biosketches. And the new Biosketch format needs to be customized to the particular grant application, and it needs to be customized to the particular situation, which means you have to get your mentors to actually work on their Biosketch and edit it appropriately for your application. This can be really hard. This is another reason you have to do these things far ahead, because they can't just send you, this is their standard Biosketch. That was true 5 years ago. That's no longer true. And if it's clear the Biosketch has not been customized to the training application, you're in big trouble, because then there's going to be skepticism that your mentoring team is truly invested in your success. Because the idea is that if they can't be bothered to modify their Biosketch to sell why they're a good mentor for you, what makes anybody think that they're actually going to be a mentor there to mentor you through the entire application? So definitely make sure that all of the mentor Biosketches are appropriately customized. And those Biosketches are going to be different in a mentoring situation than if they're applying for a grant. So just saying that they have a lot of expertise in the area, this part should really be selling what their prior mentoring experience is, where their prior mentees have gone, why are they very well qualified to be mentoring a young scientist? Okay. Now, just a few things that I would recommend here is, and this is human nature, and these are things that are just not really talked about in the directions. Somewhere on your Biosketch, list your total number of publications. So I think the current Biosketch might be your 5 most important contributions and why they're important. So this is the contributions to science narrative that you could say that, I have that listed that these are out of, just put in little brackets, out of 20 publications or out of 10 publications or whatever, so that the reviewer can see very, very quickly that what you have on your Biosketch isn't everything you've done. It might be for very young investigators, but if it's not, you really want that to be set up. The other thing that is really, really important is that you really go, it says you will also need a well-curated and accurate My Bibliography on the National Library of Medicine website. So when they took publications off the Biosketch, what they did was create the My Bibliographies on the National Library of Medicine. And I will tell you from experience, it is a pain to do these, particularly if you have more than 1 or 2 publications, and that you have to do them ahead of time. I had problems with passwords. I had problems with usernames. I had problems inputting all my pubs into that. It took way longer than I would have ever expected. But you want this to be completely up to date and well curated, because if it's not, that I will tell you that human nature is a large proportion of your reviewers aren't going to read any of the stuff you're writing on that Biosketch. They're going to look at it, they're going to click on My Bibliography and really just look to see how productive you are. So this might be the most important part, but I certainly do not recommend you blow off the rest of it. You want the entire Biosketch to be as good a sales document about your accomplishments as you can do, but particularly make sure that this My Bibliography is well done, accurate. You click on it and all the links work. Because if that's not the case, that could really, really hurt you, especially if you're going to get old fart reviewers like me who are used to NIH grant review in the old days when it was just lists of publications. Okay. Any questions up to this point? Okay. So the other thing is that, and this is really important for newly independent investigators, that somebody like me who's been doing science for 30 years, nobody's going to question my independence. I have a reputation. They're going to know what the situation is. However, especially for a lot of new clinical researchers, it's not all that unusual for you to be going in your first forays of independence at the institution where you were trained. And even if you changed institutions, as we all know, that what is independent is very, very variable, that certain positions that look independent are really glorified senior postdoc positions. And the people doing the evaluations know this. And that again, that, if you talked about the NIH rules, they'll tell you that, independence might not be that critical in reviewing a grant, but I will tell you it's, again, human nature. Study section members would way rather support somebody who really has been given a good investment by their institution, really is considered independent, than they are trying to supplement the funding of a senior investigator by funding a postdoc that's not really independent. So this is actually a really critical thing, is that if you have independent space, highlight that. If you received some sort of startup funds, that you want to emphasize these startup funds on your Biosketch under support. So where there's a part in the Biosketch where you put your current grant funding, don't hesitate to list your startup funds. I know some people are reluctant to do that because they're worried, oh, somebody's going to say, oh, I have startup funds, they're not going to fund me. That's not how the logic works here. Is that if a new investigator has been given significant startup resources, that's saying that the institution believes in the person, they are invested in that person's success, and that makes you feel more confident about having a young investigator get a good grant score.
Would the same apply to like MRDA? I mean, just to kind of show that.
Yeah, absolutely.
Because I don't know at Nemours how many. A lot of us are clinicians. How much of us?
No, but if you've shown that, like in the past, because I think it still says you list everything from the past 3 years. Mm-hmm. So if you have an MRDA to show that this is that you're getting money, and actually in that case that's even NIH money, right? So absolutely list that, even if it's over. That to show that you got it last year, that's again showing that this is something you're working on. It's really good forward progress. That I mean, the only thing that, and I mean, for a lot of if you actually have grants that are active, you need to put them on. They're required. But to be honest, that people like to fund people who are already successful. Mm-hmm. So a new investigator who has 2 or 3 pilot grants, that applicant is going to look stronger when you're going for NIH or federal money. It's not going to hurt them at all. The only thing that you always need to be careful of course, is that if, let's say, your grant you're applying for now is a continuation of this small grant funded project, you need to make it very, very clear to say, this grant will be done before the beginning of the new grant, or if it's, and I think it still asks you for potential overlap. You just explain that this was to collect the preliminary data for the current grant. This grant will end prior to the start of the new application, or you can say something on the order of that this project is not related to the current application. It kind of depends on what the situation is. But that's all good stuff. But if, for instance, like in the MRDA, that's release time to work on grants and stuff, that's, again, part of institutional support. So if you can work that in, that would be good. So looking for the support really is something that will help your application. And they will mark down applications in which investigators don't have strong institutional backing. And, this is the classic, the postdoc who's given a title to be able to submit an R01, but they have no independent space, no independent support. And the institution says, well, we'll give you that if you get the grant. I mean, that is kind of frowned upon, to be honest. Okay. Now, this is also important. Don't place unpublished or submitted papers on your Biosketch. It's against NIH rules. I mean, you can say in your little description that you're working on particular projects and you're submitting a paper soon, or if you really must, but your Biosketch is not where you put papers to be submitted or even submitted papers. It's only papers that have been accepted for publication are allowed on your Biosketch.
So it's like if it's in press, it can be on there.
If it's in press, it can be in there, but it has to be accepted. So this is just something to keep in mind, that young investigators particularly can be overly fond of putting things in preparation on all their documents. And actually funny, I just recently read a blog post, which was this rant about it. Mm-hmm. And honestly, I have seen young scientists put 7 papers in preparation on their Biosketch or on their CVs or Biosketches. And everybody who reads it, we know it's fantasy. Mm-hmm. Just because you're working on a project, you don't get to put in preparation on things. Okay, so any questions about kind of selling the Biosketch at this point?
And so you're only putting support in the last 3 years. So there's really nowhere to put, like if you had a grant 4 years ago, that sort of.
People slime it all the time.
Yeah.
It can sometimes backfire if there's nothing in the space. I have definitely seen people put on older grants on biosketches. Sometimes it doesn't kind of help because, if you're looking for a grant, then you're looking for the productivity associated with the grant. So if there wasn't like this flurry of publications in the area shortly after the grant, or there's no clear explanation why the only grant you've gotten was 4 years ago, that in some ways can hurt rather than help, because it's like, well, you got kind of launched and then you did a face plant is kind of what it looks like. So a lot of this is, thinking about the psychology of who's going to be reading these things. Okay. All right. So now we're going to talk about budget and justification. How much you have to do this sort of depends on the type of grant you're applying for. Now, from the standpoint of you need specific justification for staff and their duties and percent effort, for no matter whether you're applying for an NIH modular type application, which are the under $250,000 a year direct costs. And it's really important, again, to be explicit and realistic. That, you don't want to, if you're a new investigator and you're asking for 3 postdocs and 2 grad students, people are going to be like, I don't think so. But you also need to have enough staff on the application that they think they can actually get the work done. So it's that asking for too much or too little is a problem. And also lowballing your budget is a problem. That's something that sometimes new investigators think that, if I lowball the budget, they're more likely to give it to me. But if you are lowballing the budget and everybody reading it says there's no way they can get it done for that amount of money, it just seems naive. And also then you're going to fail because you're not going to have the money to actually do the application. Now, sometimes the budget stuff can be kind of tricky. I know for some of the F grants, the actual total amount of the budget includes the indirect costs. And depending on your institution, the indirect cost rates are very, very different from institutions. And that sometimes I've seen applications from places like, let's say, Harvard, that has an indirect cost rate of like 98%. They have the exact same total cost limit as a place that has indirect costs of 36%. And so that how much you can actually build in research supplies, salary, and so forth can really matter depending on what your indirect cost rate is. For standard research grants, R03s, R21s, R01s, the indirect costs are not counted in the budget numbers. So you do have to, again, read the directions very, very carefully. But for some of the F grants, the total budget includes indirect costs, which can really skew things quite a lot. All right, so this is something, the budget is often something you really do need to work with your institutional official about. Generally, if you're a new investigator applying for an R01, that, applying within the modular limits is usually still advised. I will say that those modular limits haven't changed in, I think, at least 15 years. And, with biomedical research inflation, $250,000 doesn't go anywhere near as far as it used to. That it is fine to apply for grants that are not modular, but you do have to be very, very careful about your budget justifications. If you are asking for, and some of the reason that is justifiable is because if you have a lot of your effort on there, and as an investigator, that's going to take up a lot of your budget. You also, though, need to be really, really careful about not overdoing the staff, because it's not at all uncommon for somebody to ask for maybe 2 postdocs and somebody to say, I don't think the research needs 2 postdocs, take it back to a modular budget. That's kind of a common thing. So this is the case of don't highball and lowball your application. Over highballing, people sometimes will over highball the application because because they think that they're expecting to get cut. But I will tell you that new investigator grants particularly often get time and amount because they want the new investigator to have the resources they need to be launched. However, if you are asking for too much and the reviewers are annoyed, they'll say, oh, we should cut that by 2 modules. But that doesn't change the amount of work that they're expecting. Mm-hmm. So, be very, very realistic, I think, is the upshot of budget type of things. And really here work one-on-one with your budget officers. It's really quite important. And this the very first R01 that I ever submitted, that it turned out that I was working with somebody. I was told from afar to work with this person on the budget, but they didn't actually submit it up the line into the central administration, and they got the numbers wrong. And this required 2 round-trip. I was still working at NIH at the time. This required 2 round-trip marathon drives back and forth between NIH and Delaware so that I could get my R01 submitted on time. Mm-hmm. And the only reason it ended up getting submitted is that this is the old days where you could actually drive it to, and I would turn in the box. Now, some of it is helpful because we now have good electronic systems, but, you want to get that budget approved all the way through your institution a week or more prior to submission, because if there's any problems, you need to fix them.
And with the modular budgets, you don't have to say each specific cost.
Okay, what you need to do is you don't have to justify every tip or tube. You do have to justify personnel costs. There are other costs you're not justifying, like you don't have to break down the rest of the budget. You also have the advantage that you can move money once you get the grant. But the actual personnel justifications are there in modular, but you're not justifying that I'm going to spend, $3,600 a year on pipette tips and tubes and gloves like you do in the non-modular budgets. Okay, so budgets are quite important. Now, the next thing that I'm going to talk about is human subjects. I will preface this by saying that I do some human subjects research, but I don't do a lot. And especially for those of you who do a lot of social science research, I don't have a lot of experience with that. But the thing is that for most of the people who are listening here, the human subjects section can make or break a grant. That they're really looking for that you have fully considered the problems and challenges. You really need to justify, you really have to show that, especially if you're studying a rare disease, that you have enough patients access to be able to get statistically significant results. That you have to really show that you can realistically get these patients to sign up for your study with the amount of money you're asking for, because clinical research can be really, really expensive. Making sure you have sufficient power based on what you're expecting to be the variance in your population. You also need to fully justify your racial, age, and gender diversity. Applications are more and more asking for this. You really can't be too obvious about this, that there are more and more pushes to make sure that everybody gets included. For instance, that you have to justify why you're not using women in prostate cancer research. You actually have to say that. You have to justify why your research on Alzheimer's doesn't include children. You need to, and this is something like I study cataracts. Most of this is adult tissue, and it's cadaver tissue is a lot of what I use. but I have to justify why I'm not studying this disease in 7-year-olds, because I'm not going to get cadaverized from a 7-year-old with this condition. So your sample sizes have to be justified biostatistically. You pretty much always have to give power analyses. If that stuff's missing, they're basically going to say, yeah, nice project, but no evidence you can get enough patients to actually make any real conclusions here. Now, that it's also really important that if your research is exempt under one of the categories, you still include that justification for the exemption. And I will say this is that almost all of my human subjects research is technically exempt because I do a lot of cadaver work. I also do a fair amount of work using medical waste. But you have to spell that out. I would also recommend that you get a letter from your IRB saying that, no, this research truly is exempt. And some of that has to do with that the people who are in the offices that submit these grants, they don't necessarily know the subtle rules of IRB exemptions. And I have, for all of my human subjects research, I have IRB exemptions signed by the IRB saying this isn't in their purview because it's exempt. And those letters go into my research office every grant I submit so that it doesn't get flagged there. Because you don't want somebody in the office saying, oh, yeah, there's humans, clicking, there's human subjects research, and then having it flagged at NIH. And this is, you do not want your grant flagged for not complying for any reason. And I will also tell you that grant reviewers are often not familiar with all of the detailed reasons for exempt research. So by you bringing it up and saying it's exempt, you're again not going to get flagged. Now, this is kind of goes into just-in-time information. It is a lot of work to get an IRB protocol approved. A lot of work, a lot of time, a lot of effort. It's also a lot of work, a lot of time and effort to get an IACUC approval approved. Now, because this is recognized, NIH does not usually require your study has received approval at the time of submission. However, there's a lot of reasons to not wait to file IRB paperwork until you know whether you're going to get the grant. One is by going through the IRB procedure, you will find out, you will basically have somebody reviewing your human subjects work. This information can be invaluable in writing the human subjects section of your NIH grant. Because if it's been through IRB, then you're not going to be sending dumb mistakes through the NIH grant process where they're going to get flagged by the reviewers of your application. Also, that even if you, say, I can't get it through IRB before I submit, start the IRB process right after you submit. And one of the reasons this is important is that I've definitely had my own IRB experiences with some of the hospitals in the CTR system. There's also, I've heard stories that other people have had, that if you're doing something where the IRB is slow, it's very, very picky, or you're doing something that the IRB does not have prior experience with, there's a real risk that you won't get your IRB approvals done when just in time, let's say you get a great grant score, they ask for just in time to be able to get your IRB approved in that. It's a very short window for just-in-time information. It's like 4 to 6 weeks very often. And if you can't get your IRB realistically done in that time, then your grant is going to get delayed. And in some circumstances, it can even not be funded because there's a lot of grants that get funded on end-of-year money. There's lots of grants that get funded on other purposes. And so that one thing is that once you've gotten a type of project through IRB, getting modifications through it is nowhere near as much of a problem. So, be really careful about this. And I know there's a lot of people listening who are doing IRB type research. And, I would be worried if you decide to not go through your IRB until you get a just-in-time statement. A lot of the things that I said for human subjects are also true with animal subjects. Animal subjects is something that I have never submitted a grant that does not use a lot of animal subjects. This is something I'm very familiar with personally, is that you have to fully address the 5 points in the application. And you should note that sample size must be fully justified biostatistically. This is, again, power analysis, or if you can't do power analysis or you don't feel it's necessary for some reason, you have to explicitly say that. Now, some things that when they say provide a detailed description of the proposed use of animals in the work, identify species, strains, ages, sex, and number of animals, that you have to fully justify why you're only using one sex if you're only using one sex. Now, this is a case is that, all right, if you're studying, again, prostate cancer, yeah, only male mice have prostates, but you should say that. But there's a lot of studies that basically omit females because of variability, because of the estrus cycle. That is not an adequate justification for excluding from the study. So you have to really justify this for scientific reasons, not necessarily just for convenience. And also that cost reduction, like in this case, if your results are more variable, you're going to have to do more animals to get power. That is not an adequate reason to exclude females. So you have to be really careful about that. Now, to justify that you're using animals at all with species and the numbers, and you have to provide a rationale for the selection and numbers here. Now, this is, again, talking about, the sheer number of animals. Now, one thing that if you're working with genetically modified organisms, you need to include all the animals that will be generated in your study, but not used directly. I know that in my work, it's often a case where only 1 in 4 animals are usable in my study because of the genetics involved. I can't just talk about the animals I'm using in the study because of the other 3/4 of the animals are going to be byproducts of that study. And you need to be including those animals here. And one thing about this is that there's a new requirement about fully explaining and validating your scientific resources. This is a case where you have to really talk about what are you doing to prove that you really have the mutation you say you're having. If you're saying you're working in a particular genetic background, what experiments are you going to do to validate it's the correct genetic background? That it's an entire new thing that was just added for the most recent rounds of application is how are you justifying your scientific resources? That I don't have a separate section in this handout about this, but this is also something that if you're doing a lot of cell line work, you have to explain what ways you're characterizing that cell line to show that you're not really working with HeLa cells that have contaminated your culture. So there's every few passages you have to go back and do the genetic profiling of your cell line to show it's still the cell line you think it is. So it's, again, validating your scientific resources, something NIH is getting more and more sensitive about. This is true for mice. It's also true for cell lines. Okay, so provide veterinary care of the animals involved. You don't say, yeah, there's a vet, or yeah, there's an animal facility. You want to be very specific here. You want to talk about what are the vet, whether there's part-time vet, full-time vet, who that vet is, what their training is, what is their experience doing animal research. So that they want a lot of detail here. Also, describe the procedures for ensuring discomfort, distress, pain, and injury will be limited to that unavoidable. That they want details, that you want to be specific, you want to give drugs and doses, that really talk about how you're doing, if you're doing surgical trials, how you're doing anesthesia, so forth and so on. They want specifics. That similarly with euthanasia, So you want to be very, very specific here. You need to refer to the American Veterinary Medical Association's guidelines on euthanasia. You need to include scientific justifications for not following those recommendations, and you must be explicit. Now, this is all of the things that I said about being careful about just-in-time information with IRBs is also true for IACUCs. That pretty much all of the information that they're asking for here in the research plan related to animals is also the same sorts of things you're going to need to address in the IACUC approval. So if you go in and submit the grant without IACUC approval, you're again submitting things that have not been through your IACUC. There's always a chance that the IACUC would have found mistakes or things that are not covered adequately, or maybe the drugs or the anesthesia or the euthanasia methods aren't allowed. You want that flagged by your IACUC, preferably not by NIH. So it's usually better to at least get your IACUC vetted by your vet, looked at by your vet. And I also would not wait until to do the paperwork for just in time for the same reason, is that, again, IACUCs can be slow just because they might only meet once a month, or they can be picky, or particularly, again, if you're doing something non-standard, that your IACUC may ask for so much more information. And if you can't get that IACUC approval within a month after the grant score comes in, it's again going to slow and can even lose your award. And I actually had a friend of mine who almost lost his R01 award because he wrote his animal section in a way that annoyed a reviewer, and they flagged it. And it went all the way to the highest levels of animal review at the NIH. And he ended up getting his grant, but he had to probably do 10 times more paperwork. And he was on pins and needles until he finally got things approved. And it was a pretty simple protocol. It was just had to do with, that if somebody flags something, then you're in trouble. And this is also, and one thing that I want to say about human and animal approvals, and this has to do with being very specific and making sure, again, you don't leave these things to the last minute. That, let's say you have a funded grant or have written this section before. You can't leave this to the last minute. Make sure you read it far enough ahead of time and really read it carefully, because it's not at all unusual to see somebody stick their animal or human subject sections from a prior grant where they're talking about different mouse mutations or different human diseases. And it's just that somebody got busy. They're like, oh, I've written the human subjects, I've written the animal subjects, oh, I'll just lift it and stick it in the new application, and never checked to see whether or not this animal subject section or this human subject section makes sense for the grant being submitted. And this is very, very common, is that where I have seen people talk about in the animal subject section research or techniques that are not proposed in the grant. And I have seen this section lacking techniques that are approved in the grant. But the one about the wrong mutations or the wrong diseases, that is really, really sloppy. And it's just something that nobody can really afford because it will flag your grant. Also, vague is bad. Again, some people might be able to get away with vague if they're well known in the field or if the institution is very, very well known. But few of us can really assume that we're going to get the benefit of the doubt because we're so famous. So be as specific as possible. And sometimes people will get away with sloppiness or vagueness and then suddenly get blindsided. And I will just, as full disclosure, I tend to be a very, very picky reviewer about these sections. And I have flagged a lot of people's grants just because that it's also it's a lack of respect, it's sloppiness, there's lots of problems with it. So, definitely address these things early. Okay, so any questions up to this point? All right. So the last.
I have a question.
Go ahead. Sorry.
Yeah, I have a question concerning exceptions from IRB rules.
Hmm?
I mean, if we are not using directly animals, but if we are working with blood samples obtained from clinic, do we still need IRB approval, or it's some kind of exception? Can you
Okay.
please explain?
All right. So I will tell you that I would definitely discuss this with your IRB. Generally, blood samples from the clinic, it depends on whether they're identified or de-identified. So if you are able to get de-identified samples, like if you just need random Joe person's blood, that or like leftover units of blood from the blood bank that expiration or something, that's usually de-identified human subjects material, and it is considered not human because it can't be referred back to a human. However, I would still recommend, if that is your situation, to ask your IRB for a letter of exemption so you have it. If there is any possibility of the and also in that situation, I would also write in a specific provision if the identifications come to you by accident. And I'll say this from my own experience, is that I work on a very, very rare disease, and I get de-identified clinical specimens from people, from surgeons across the country. Sometimes the charge nurse, they say, oh, here's this specimen, send this to Melinda Duncan. Well, they won't erase the label on the sample container. And I have gotten people's names and codes from clinical information, even though it was supposed to be de-identified to me. So I have written into my thing, what do I do if somebody inadvertently tells me who the sample was from? And you can imagine, like a unit of blood, there's lots of things that could happen with. It just covers yourself about how you will basically say, I'm going to destroy this information the second I get it. That if it's a case where you will, that it is possibly identified, let's say you're working on a study where you might not know who the people are, but you'll know that this is patient 2362, and somebody in the larger study can take patient 2362 back to a real person. You may need to get IRB approval, just so that, and it's called expedited review, where you just have to show that you have taken adequate care to make sure that stuff isn't identified. And I just say this from experience, is that you can get stuff by accident that you don't want. And like, I flipped out when I got these specimens from this surgeon in Ohio, where I had people's names and mailing addresses. And you just want to make sure you set things up in such a way so you don't get in legal trouble if something inadvertent like that happens.
Yeah, and just to jump in, I would recommend, the first step would be just reaching out to your IRB and asking for a review for research designation, and then they can actually tell you, we can review this expedited, or, it's exempt, but that way you're giving them all the information they need to tell you what additional information needs to be submitted.
Yeah, I would very much concur. And if they say it's exempt, we don't need to review it, ask them for a letter with a signature of the IRB director saying that is considered exempt. Because that on more than one occasion, like we've had a new person in the grants office who wasn't familiar with my work or how it could fit with exemption. And you don't want your grant to get held up through your institution because there's some question about whether your research is exempt. If you can just send the IRB letter in when you submit your grant, it just removes a lot of confusion. Does that answer your question?
Yes, thank you.
Okay. Any other questions about that?
Yes, I have a question. If the project involves multiple institutes, and then each institute have a different IRBs, how do you recommend for the IRB approval?
Okay, so I will say that this is a little bit beyond my pay grade. That generally the work that I've been doing is that the IRB approval has been through the institute, that there's been IRB approvals in both the place sending you tissue as well as IRB approvals in the place that the grant is coming out of. I know generally the University of Delaware is that if from their perspective it's exempt research, that's as far as they need to take it. There are moving target rules on this related to IRBs and multi-institutional awards, and they're trying to make it simpler, but I don't know the details. I think the best bet would be you should contact the IRB at your primary institution and go from there. That I don't want to give you any advice that could be wrong, because I know that this has been a moving target in the past couple of years.
All right, thank you.
To chime in, I would agree with that. In my experience, generally the home institution needs to apply first, and then once that IRB is approved, the other institutions that we work with then have to also kind of approve the protocol as well. But your IRB should be able to help you make that decision.
Any other questions? Okay. So the next section is resource and data sharing plans. This is something that I think people are getting more used to, but it's a really easy thing to omit, and you will get flagged and in some kind of serious, I don't want to say trouble, but you'll definitely have a lot more headaches than you need to. That this is a requirement that started maybe 4 or 5 years ago, that basically any resources for the scientific community, this can be cell lines, this could be genetically modified animals, this could be clones, this could be databases of some sort of information, that if you create these resources or you're collecting a lot of data, like you have a lot of human subjects data or a lot of genomics data, after publication, you must share these resources and make the data available publicly. That you need to write a section that describes, what would be how you would decide whether to send somebody a genetically modified mouse or a cell line or so forth. One thing to keep in mind is that if NIH funds the creation of, let's say, a new mutant mouse or a new cell line, you are obligated to share that with other people. Now, you can have people sign material transfer agreements. You can require people to show documentation that they have the facilities to take a mutant mouse in under the correct rules, but you are required to share these resources. There's a lot of scientists who still don't quite get that, and they think that, if they made the resource, they get to decide what's it used for the rest of time. That's actually not something that they have much of a legal leg to stand on with. And this research and data sharing plan section is basically explains under what criteria what resources are you creating, and under what criteria will you share that information. Now, most institutions have a boilerplate about this, about legal aspects of data resource transfer in relationship to MRDAs. I know UD there's a uniform material transfer agreement that a lot of institutions use, but you start with the institutional legal boilerplate and then you customize it to your project, because every single grant will have a different set of new resources. And admit this at your peril. Definitely you want to include this section unless you really aren't making any new resources. I would say probably 90% of grants do create new resources in some way, shape, or form. But maybe you're in that 10% that isn't. And then the very last section that I would also be very careful about is the biosafety section. I've seen a lot of grants admit the biosafety section because I think people, for some reason, think that this is only if you're working with Ebola or something. Biosafety is everything that's at biosafety level 2 or above. This involves human clinical material. This involves lentiviruses. This involves retroviral vectors. Anything that has a placarded lab. If you're working with human tissue, there's biosafety issues associated with that. And it's just a case that once you've written a section, then it's there and people will be good with it. But you can't omit the section if you are doing that kind of research. And, please don't forget it. Again, there are many grants that go through that have biosafety issues that nobody flags. But again, if you get a picky reviewer, you just don't want to go there, right? You don't want to have to backfill things so that your application is flagged as non-compliant with the rules. And then the very last section that they now have this whole thing about making sure you're validating all of your scientific resources. And this is validating your cell lines, validating your mouse mutants. If it's something that you're using a reagent, you have to explain how it's been validated. Validating antibodies, so forth and so on. So I think with that is I think that the take-home message from this section is please make sure that you don't take all of the associated verbiage of the application. Don't get lazy with it. Don't discount it. It can be equally important for you to finally get your application or get your grant. And you don't want people to get the impression that you're sloppy, because if you're sloppy on a grant, maybe you're going to be sloppy in science. That's the implication about this. Some of it is once you have it, just make sure that you've edited it, make sure you've updated it for each new application. I think that's it for right now. Is there anything, especially for the Moore's grants officer, that they want to chime in about? If he's still on. He might not still be on. Okay. Any questions for at the end of this, or are we good? Getting lots of quiet. So I'd like to thank everybody for coming. And I don't remember when the next one is, but it's sometime in April. So see people in a month. Bye.
Thank you.